Recently, Otzi the Iceman has given microbiologists a chance to look into what the human microbiome of the past may have looked like. Otzi was preserved in ice for multiple millennia at a time, and even though he died completely isolated, he did carry, most likely, a similar microbiome to other people of his time. 7 years ago, it was discovered that Otzi had a Lyme disease infection, which is caused by a bacteria known as Borrelia burgdorferi, and it's most commonly spread by ticks. But more recently, new research has been done into specifically Otzi's microbiome, coming out just a handful of weeks ago, actually. Scientists have linked Otzi's Lyme disease infection to several of the conditions that he had, including his arthritis and possibly his atherosclerosis. While Lyme disease has not been directly connected to atherosclerosis, viral and bacterial infection have been proven to significantly inflame the arteries and cause a vastly increased risk for atherosclerosis. Otzi lived at a time where people had very low-risk diets for heart plaque, and very few people of his time ever developed this condition. So it does hint at underlying genetic causes or his infection that he carried. But just this year, a new technique has been used to analyze more microbes on Otzi. While we knew that he had carried a handful of yeasts on him as well as the Lyme disease infection, we did not know the full microbiome of Otzi, and we still do not have a complete understanding of what his microbiome looked like, but recently, new technology called aDNA analysis, also known as ancient DNA analysis, has allowed us to look into what his microbiome might have looked like. Some of the bacteria that we identified through aDNA analysis has later come back and been proven to most definitely be modern human contamination, rather than actually a part of Otzi's microbiome. And some bacterial species, specifically Staphylococcus, we're just not sure. We're attempting further analysis to see if it matches modern strains, but even that's not necessarily proof that it came from modern contamination. Some bacteria found in Otzi are completely extinct in the modern day, and that proves that it is most definitely not modern contamination, but unique to him. One of them is Treponema succinifacans. It is actually from the same genus as Treponema pallidum, the cause of syphilis. However, succinifacans is nothing like its cousin. It's far from pathogenic and is highly beneficial to the gut. It helps people digest fiber, and a recent study by Harvard Medical School found that 0% of modern humans carry it. Modernly, our main microbe for fiber digestion would be non-pathogenic strains of E. coli. However, Otzi had a vast array of several microbes that specifically aided him in fiber digestion. The new analysis also found an H. pylori infection on Otzi, which is really nothing surprising, as in hunter-gatherer populations, or at least modern hunter-gatherer populations, nearly 90% of the population carries this microbe, whereas in industrial society, only around 30-40% of us at most carry it. In both societies, around 80% of carriers are completely asymptomatic, and the other 20% develop conditions like gastritis and ulcers. But what's interesting is stomach cancer is only linked to H. pylori in modern Western civilizations. Hunter-gatherer societies do not see this complication, even though they see other complications at the same rate that we see them. Most scientists think it's most likely competition, because they have a very vast, diverse microbiome. They have competition within their microbiome. But one of the yeast identified on Otzi 16 years ago has, interestingly, been recently found to be more abundant than it was initially, which hints that this specific yeast is not only good at surviving in cold environments such as the glacier, such as the museum's storage center, but it also hints that it's possibly able to feed on the materials that they're using to preserve the Iceman. Otzi was perfectly preserved in ice for thousands of years, and he was a prime candidate for aDNA technology. However, the hope is in the future to use this technology for remains that are significantly less preserved. The hope is to possibly map the complete evolution of the primate gut microbiome, possibly the gut microbiome of animals to the extent that we can. It is a significant challenge to map the evolution of bacteria themselves, and if we were able to map the evolution of animals' microbiomes, that would very significantly tell us a lot about the evolution of bacteria itself as well.